I'm going to talk today about the National Cancer Registration and Analysis Service. So that's who I work for. We're part of NHS Digital. And I'm going to talk about the molecular biology and why molecular and genetic data is so important in cancer, and particularly in GISTs. So just a disclaimer here, I'm a scientist, I'm a geneticist, I've got a PhD in cancer genetics, but I'm not a medical doctor, so I'm not an expert in the diagnosis or treatment of GISTs. I just know a lot about DNA. So just a quick survey around the room. Has anyone here heard of the National Cancer Registry? About a third, I think. Okay, that's not bad. So I'm going to tell you a bit about who we are and what we do. As I've said, we're part of NHS Digital. So the overarching department is the National Disease Registration Service. And that's split into two parts. So there's the National Cancer Registration and Analysis Service, and then we've got the National Congenital Anomaly and Rare Disease Registration Service. So from the perspective of today, we're talking about the cancer registry function. And my role within that is to manage the genetic data program within that. So as the National Cancer Registry, we collect and quality assure and analyse data on anybody who's been diagnosed with or treated for cancer in England. And we have a legal statute, which is a very, very dull read, but it's quite important. But that enables us to collect data on all of you guys without explicit patient consent. So we're trying to get the word out there that this is what we're doing. And the reason that we need that legal statute is because we need to be able to calculate the cancer statistics at a population level. And if we had to get consent from everybody on an individual basis, it would be a logistical nightmare. And also, it's probably not what anyone wants to hear when they've just been diagnosed with cancer, to be talking about, oh, can we do this with your data? So this goes on behind the scenes, but we actually want people to know that we do it. And one of the important outputs we do are these national cancer statistics. So if you go on the Cancer Research UK website and you want to know how many people have had GISTs or what the survival rate is and so on, that's all there on the Cancer Research UK website, but those statistics actually come from us in the National Cancer Registry. So that's the only way we can get population level statistics. So that's who we are. So going into some statistics for GIST cancers, these are the stats from 2016 to 2019. We're just about to release the 2020 cancer statistics, but because they haven't been officially released, I can't talk about them yet. So 2019 is the most recent year for which we've got complete cancer stats. So the column to look at here is the blue one, the ASR, that's the age standardized rate. So that tells you out of 1 million people, that's how many have had a GIST. We can calculate the average, and over 2016 to 2018, there are about 841 GISTs per year. Now in 2019, it looks like there are a lot more, that's 975. There aren't really more GISTs in 2019. What has happened is the way that they're classified. So Dr. Ali, you showed effectively this same slide, didn't you? So if we look at the top, these ones with the very tiny ones with the low mitotic index, they used to be classified as benign. So because we've only got the legal permission to record cancerous or malignant disease, we couldn't record any of these. But now the World Health Organization and the Royal College of Pathologists has changed the classification. So effectively, these are all counted as malignant diseases. So from 2019, we've been able to collect data on all GISTs, even the very, very tiny ones, which have an infinitesimally small risk of doing any harm. So that's why it looks like the number of GISTs have gone up for 2019. They haven't, it's just the way they're classified. So I'm going to talk now a little bit about some of the science behind DNA and genes and why that's important in cancer. So probably everyone here knows that we're all made of cells, and every living organism is made of cells. So here's an amoeba. He's just got one cell. The nematode worm has exactly 959 cells, not one more, not one less. And a human being has about 10 to the power 14 cells. And this particular human being is Sir John Sulston, who did a lot of work on the nematode worm and could classify exactly where each of those 959 cells came from. It's pretty geeky work. Well, you'd be sacked. You wouldn't be a good worm scientist if you couldn't count beyond 820. But Sir John Sulston actually did a lot more than just counting cells in nematode worms. He was one of the driving forces behind making the human genome publicly available, because in the early days there was a lot of sort of corporate interest and particularly in America, trying to put patents on genes and, you know, keep some of the information. So John Sulston, he passed away a few years ago, but he was a real trailblazer in saying, no, this belongs to the whole of humanity. This does not belong to a corporation. So that's very much why we've got free access to genetic data. So he's a bit of a hero. So yes, cells are really important, and the brain of the cell is the nucleus, and that's where all the DNA is, and genes are made of DNA. So within the nucleus of each of our cells, we've got about 2 meters of DNA, all curled up very tightly. And the protein synthesis, each gene is effectively a recipe for a protein, and the proteins are made in the cytoplasm of the cell. So that the DNA is kept separately, the nucleus is effectively your inner sanctum, and you want to protect the DNA from all the nasty business that goes on in the rest of the cell. Here's the human genome. So it's 3 billion base pairs of DNA, 20,000 genes, and nearly 99.8% sequence identity between any two of us in this room. And this picture is from the Wellcome collection. It's a bloody good read, actually, this genome sequence. And here it is divided up into all its volumes by chromosome. If you've got a spare 100 years or so and you're in Euston Station, just pop over the road and you can read this for yourself. It's fascinating. We're quite similar to other species, so there's 98% sequence identity between human beings and a chimpanzee. And here's a lovely young chimp, and here's one of my favourite examples of the human species, who's my cheeky little nephew, who's quite similar to a chimpanzee, and I think the resemblance is quite striking here. And the reason for that is that there is 98% sequence identity. I suspect in his case it's about 98.2% actually. I might be, yes, yeah. Especially as I make him birthday cakes like that. What you can't see on that, he said, I want a volcano cake with red lava, a green T-Rex with black spots, and a brown poo. The poo was down there. I've slightly cut that off from the cake because it needs some explanation. But yeah, it's the first time I've made a poo out of sugar paste. I quite enjoyed it. So all human beings are a little bit different, as you can see by all these children. And the reason for the differences between us is because we have small variations in our DNA. This was some DNA sequencing that I did as a PhD student. It was a good few years ago now, so we do sequencing a bit differently now. But you can see where I've highlighted, the blue peaks are the C and the green peaks are the A, and you can see that they overlap in those two places because we've got 2 copies of our genome, one from mum and one from dad. So this is just a normal variation in DNA. This one's not a particularly important variation, but you can just see it there. And then we also get variations in our cell types. So every cell has got exactly the same DNA sequence in its nucleus, but yet different cells behave in different ways. So your cells in your eyes are doing a very different thing from the cells in the liver, for example, but they've all got the same DNA sequence within them. So why is that? Well, the reason is that in different cell types, different genes are being switched on and off. So your sort of bread and butter genes, your housekeeping genes, they're switched on in every cell of the body. They're really important ones. And then you've got the tissue-specific genes, that your liver enzymes would only be switched on in the liver. And the gene for hard keratin was just switched on in the fingernails, and so on. So if we look at this cell here, that's where gysts are arising from. It's that part of the embryo. So the thing to remember about cells is that the behavior of a cell depends firstly on the DNA sequence of its genes, and secondly on which genes are being expressed or switched on within the cell. So now we need to think about, why do some cells become cancerous? And the abnormal behavior of cancer cells is because we've got particular DNA sequence changes in those cells, and some genes in cancer cells are being incorrectly switched on or off in the cell. So that's why cancer cells behave differently. It all comes down to the DNA. I rather like this quote at the top of this slide. Nothing in biology makes sense except in the light of evolution. And this was a chap called, I'll probably pronounce this wrong, but Theodosius Dobzhansky. And he's what, sorry?
It's easy for you to say.
Easy for me to say, yes. I did have to rehearse that bit. So let's just think about evolution and how that works. So we're taking our old friend the amoeba, our single-celled organism. If that amoeba gets a mutation in its DNA, and that's a disadvantageous mutation, the amoeba will die, the mutation will disappear with it. If the amoeba gets a neutral mutation, that will just be retained for the life of that amoeba, and it will go into its offspring. If we get an advantageous mutation, that amoeba will have lots and lots of little amoeba babies, and over time that mutation will become more prevalent in the population of amoebas. So now let's have a think about what's going on in a human body cell. So it's the same sort of thing. If you get a disadvantageous mutation from the point of view of the cell, in, say, a cell in your fingertip, that cell will die and the mutation will disappear with it. If you get a neutral mutation, like if one of your liver cells develops a mutation in a gene that makes something important for the eye, that liver cell's not going to care, is it? Because it's not using that gene anyway, so that cell will still be perfectly happy. But if a cell in your body gets a DNA mutation in something like a growth factor that makes the cell grow and divide, then that cell will have a survival advantage. And that cell will grow and divide a bit more and make a clone of cells. And if one of those cells develops another mutation that makes it grow faster, you're well on the way to cancer. So cancer effectively is Darwinian evolution at the level of the cell. And you can see this here. Here's Charles Darwin. Here's his notebook from 1837, and you can see he's written at the top, "I think," and he's shown how he thinks all the species branch off and evolve. And then about 10 years ago, they sequenced a kidney cancer, and you can see here, that's the first mutation to happen in the kidney cancer, a mutation in a gene called VHL, which, as everyone knows, is on chromosome 3. And then further on, you get these other mutations. So you can see really that what Darwin's saying and what's happening in this kidney cancer is exactly the same. You can see this evolutionary tree. So cancer is a micro-evolutionary process going on within our bodies. Okay, so that's cancer genetics in a nutshell. So how do we treat cancer? So the traditional ways, cut it out, burn it out, or poison it out. And it's quite primitive. I don't know if we've got any surgeons in the room. No, we haven't. That's a shame. I like to tell surgeons how primitive their techniques are. Disappointing. Chemotherapy and radiotherapy kill normal cells as well as cancer cells because they kill anything that's dividing quickly. And that's why these treatments, particularly chemo, have such brutal side effects. It's horrible stuff. So we need to get gentler and more specific ways of treating cancer. And this very much ties in with what Dr. Ali was saying. So here we've got a patient with a tumour. And what we can do now is look at the DNA within the tumour and see exactly which genes are mutated in that tumour. And we can profile what mutations are in there. And on the basis of the mutations that have happened in that tumour, we can prescribe a targeted treatment. So this is where imatinib comes in. So here's a cell. This is a cell in the intestine, and it's got a KIT receptor on it. And the ligand, the docking, the key for KIT is a substance called stem cell factor. So in a normal cell, when stem cell factor binds to KIT, KIT sends a signal to the cell nucleus, and that switches on growth genes. And when stem cell factor has gone, KIT stops signaling, so the growth genes are switched off. So that's what's happening in a normal cell. Now in a GIST, in a cancer cell, the KIT gene has a mutation in it, so therefore the KIT protein is abnormal. So that is constantly switched on, even in the absence of the stem cell factor signaling. So these growth genes are permanently switched on, and that's what causes the cancer. And this is why we can treat GISTs with KIT mutations with a targeted therapy, such as imatinib. And imatinib, it's a really important drug because imatinib was actually the first targeted cancer therapy ever, and it was developed and launched in 2001, and initially it was a treatment for chronic myeloid leukemia, so it's sort of been repurposed for treatment of GISTs. I think the first time I knew that imatinib was used to treat GISTs was when I was a lab-based researcher. I was at the National Cancer Research Institute conference. I think it may have been here in Liverpool, and I met Judith, and she told me about imatinib being used in GISTs. So, yeah. This next one is a bit random. So, what we've had before is a KIT that's been overactive just within a particular cell or a particular set of cells. Now, this is an example of what happens when KIT is underactive. And this is an inherited mutation, so it's in every cell of the body. So the cells that make the intestinal cells that cause GISTs and the melanin-producing cells in the embryo, they're all in a part of the embryo called the neural crest, and those cells have to migrate to the right places. So if KIT is underactive, it's lost its function. And what this means is that there aren't enough of these pigment cells to pigment these this baby and these animals properly. So that's how piebaldism happens. So things like piebald horses have obviously been bred for that sort of thing, because as humans we like the appearance, we like the pattern. But yeah, that's sort of the opposite problem. So it's the same gene, but it's been affected in a different way, and this gene is mutated in every cell of the body. So obviously this is a pretty benign issue, isn't it? It's just a difference in coloration, and as I say, we sometimes prefer horses to have this colour. And it's the same with cats as well. When they have their little white bib, that's the same sort of thing. There just aren't enough of these pigment cells to go all the way around because they're growing a bit more slowly. So that's just some random science for you, because that's the way us scientists like to roll. Okay, so what am I actually doing at work when I'm not thinking about some piebald horses, which I don't tend to do very much at work. So my job is to collect genetic data from all the labs around the country. 7 years ago, I was trying to do this single-handedly. Now I'm glad to say I have a team of people helping me with that, which has made it easier. So in the cancer registry, we collect all sorts of information that are generated as part of a person's medical record, as part of their care plans, the treatment that they're on. And we bring all that into the cancer registry. All the hospitals send us copies of the data. Here's our big database. And within the molecular and genomics team, we've trained up our fabulous cancer registration staff, or a subset of those, to deal with DNA data, molecular data, and that includes the GIST data. And I have to say, these cancer registration staff are absolutely fantastic. They're not scientists, they're admin grade staff, really highly trained, but you know, they're not paid that well. They're just very normal people, and they're doing this amazing stuff and registering some really complicated molecular data. So I always want to give a shout out to them, because I'm just so proud of them. So we get all this information into the cancer registry database, we bring it all together, and then it goes into the analytical part of our organisation, and it's by the time it gets in there, it's all anonymous. And then we can use the data to produce our cancer statistics and new insights. So we've used our molecular data to look at how many GISTs are receiving these molecular tests. And you can see that over the years, more GISTs are being tested, so this is good news. We really want them all to be tested. And we can see here that the yellow, that's the Birmingham lab. So Birmingham are doing the vast majority of the GIST testing, and that's not surprising, because the lab in Birmingham is huge and does a lot of this sort of cancer testing. So overall, we've got just under 40% of GISTs given molecular tests. 38.2%. So it's not enough GISTs are being tested. We know we have got a bit of missing data in there, so the real figure will be slightly higher, but it's still not enough. So who's more likely to be tested? Well, the main thing is that younger patients are more likely to receive a molecular test when they've got a GIST. So we're counting here younger in the below 44 age bracket. And then as you get older, you're less likely to receive a molecular test. And obviously the molecular testing is really, really important, because if you've got a KIT mutation, if you've got a PDGFRA mutation, really people like Dr. Ali need to know about it. They need to know what that mutation is to be able to give the best targeted treatment. So it is really important that everyone would just get a molecular test. And this is the first time, so the work we're doing in the cancer registry is the first time really we've been able to look at this on a national level and say, well, who is doing this testing and who isn't? And we can divide that up by which cancer alliance is in charge of the area. So the two at the bottom, Thames Valley and South East London, I've put them in a slightly different colour because we know that the issue there is that's two particular labs who are not yet sending us data, so that's not representative. But you can see really across the country we've got a lot of geographical disparities in terms of the percent of GIST patients who are getting these molecular tests. So one of the things that we're trying to do as a cancer registry, not just for GIST, but for other types of cancers as well, where molecular testing is super important, we're trying to get this information out there and sort of name and shame the cancer alliances whose testing rates aren't so good. And I think you know, this is something we've only been able to do really for the last few years, because I only started in post in 2015, so 7 years ago. So before that, we had no genetic data in the cancer registry at all. And clearly it's taken a long time to sort of start from a blank piece of paper to get to this level where we can sort of do these statistics. So this is a bit of a game changer. So we're going to try and put this out there and, you know, name and shame some people for not testing. And actually, just a bit of a boast about the cancer registry. I think we're the only one in the world that is recording molecular data to the degree of specificity that we do here in England. So even in America, they're recording bits and bobs of molecular data, but it's not on the same scale of what we're doing. So I think we're proud to have the best cancer registry in the whole world. And then we can delve into the mutational status of, so these are all the GISTs from 2016 to 2019 inclusive that were given molecular testing. And as you can see, about 3/4 of them do have a KIT mutation. These are the ones in the orange. The dark blue, 11% have a PDGF-RA mutation. Yeah, sorry, the green have the PDGF-RA mutation, that's 10%. We're not quite sure in 3%, one or other of the tests failed. And then these in the dark blue, these are the wild type ones here. And then sometimes further testing is done on the wild type. So wild type just means there's no mutation in KIT, there's no mutation in PDGF-RA, so the two most common genes are not mutated. So of those wild type ones, probably about half of them are not given any further testing, which is not great because, you know, really it'd be good if they were all given further testing. These ones were given some further testing, and mostly the gene that was tested was BRAF. And we've got a few at the top where the SDHB gene is not being switched on properly. That's what underexpression means. And then there's a couple at the top with a BRAF mutation. So those are much rarer mutations, but it's important to find because, again, it does affect the treatment. So, yeah, in summary, with the National Cancer Registry, we're doing this work with genetics data, and we found that only 38.2% of GISTs are given molecular testing, and there's big geographical disparities. So, yeah, big thank you to all my colleagues, particularly the cancer registration staff, and to one of our analysts, Andrew, who produced this data for me. And thank you very, very much for listening.